We have started with a chapter called as biological classification, and we have started the first part of the lesson. And what we were discussing was we were talking about the different types of scientists and their contributions. Which one? Here itself? Thinking about botany? Okay, anyway. So like we were talking about the different scientists and their contributions, and what we said is what are the important points to be studied from examination point of view, right? Prottection scientists or kekhonman kitab ase, some books that they write. So these are the points that we need to study. Today we'll be continuing with that part itself. Once we complete talking about the contribution of the workers, we'll discuss about the kingdom system. Kali kiman number loke likhisa? Yeah, it was number 6 I think, right? So today we'll be talking about the next contributor, likhiba number 7. Right? Talking about Karl Marx. So detailed questions from all these sections, no, it's not seen, like it's not very commonly asked, but there are two things that you need to remember. There is a word called as serology when we are discussing about Karl Marx, right? So Karl Marx he has given the importance of serology in the method of classification. Means what? Every scientist was trying to classify the organisms, and while classifying they have taken certain basis, certain points as a basis for classification. Someone considered morphology, someone considered all the points, maybe someone considered even phylogeny. So Karl Marx jetia classification korisile, what did he talk about? He talked about serology, and talking about serology, he tried to show the similarities and the evolutionary line between the two organisms, right? Serology mane ki? The protein structure, each and every cell. If you are talking about a cell, what is the building block of a cell? Ekdam basis ki proteins, right? So what he has done, he has tried to study the protein structure of the cells of the organisms. Suppose he has taken organism A and an organism B. Organism A serological study or the cell or G protein ase, what is the structure of the protein in the organism A and what is the structure of the protein that is seen in the cell of organism B. Both of them he has taken it into account, and then he has started comparing. What did he do? If he saw that there are more points of similarities between organism A and organism B, he concluded that most probably maybe organism B has evolved from organism A or organism A has evolved from organism B. So ultimately what we need to know is it is on the basis of protein structure that this classification was given. So you don't have to remember protein structure and all those. Question ata ahise, name the scientist who has done a classification on the basis of serology. So just that word and the name of the scientist, nothing in details, right? It is also believed that we often talk about that man and chimpanzee relationship, right? We talk about that as if we are evolved from the chimpanzees, right? So one point of study that proved that statement was also serology. Right? So men or human beings or protein structure porisile, and he has studied about that protein structure of chimpanzee also, and ultimately they have seen so many similarities between the two cells, and ultimately they have concluded that maybe human beings have originated from chimpanzee. Iman details ki to nalage, right? So as far as writing is concerned, ako ekita points likhi dibo laibo no etia. Likha. Ne tumalke sobe likhisa niki? Nai likha no? Okay, June likhisa, just wait. Another thing is, as I am taking the class, not necessary each and every point, each and every sentence that we will write, okay? As I am discussing, as I am dictating, there might be certain points which you will note it down. Right? Moi koi thakuteo ketaman points thakibo pare jonkhini ki tumalke likhi loba, right? Karon prottektu point likhibo lagile to bahut samoy lagibo, right? And at the end of the lesson and at the end of the chapter, obviously PDF you are going to get. This chapter you have got the entire PDF, but from the next chapter itself, PDF is something that you are going to receive only after the completion of the chapter, right? So what you do, take the notes in the class, write certain notes that I speak, right? And at the end of the lesson, compare it with the PDF that you are going to get it. It will be almost the same content, kintu okoman eta duta sentence beleg hobo pare, right? Okay, so show the importance of serology. What is serology? Likha study of protein structure. What is phylogeny? Evolution. In very short terms. Okay. So, after this was point number 7. After this, we need to talk about, since we are going in a sequence. So, after Karl-Menz, we need to talk about Whittaker. But Whittaker's contribution we will study a little later. After Whittaker, June asile, we will be writing about it. Next, likha number 8 boli likha son. Number 8, R.H. Whittaker. Details about Whittaker, we will write it later. Number 9, Karl Woese. Okay. Points about Karl Woese. Whenever you hear about this scientist, you need to remember that he was the one who has given the 6 kingdom classification. As we have spoken about it in the last class, what we said is the classification systems from time to time it got advanced or it got evolved, right? It started from the 2 kingdom classification and gradually it was started being better because there are certain demerits with the respective classification. So, from 2 kingdom classification onwards, as the scientists kept on working, we have now the 5 kingdom classification system which is most accepted. But apart from the 5 kingdom classification, we have another one that is called as the 6 kingdom classification. So, what are the points that are asked from the examination point of view? The first point, who has given the 6 kingdom classification? It is Karl Woese who has proposed the 6 kingdom classification. Next point, on the basis of what? On the basis of ribosomal studies, right? What is a ribosome? What is the structure of a ribosome? This is the content that we are going to discuss in cell, right? Cell, the unit of life, we have a chapter like that, and at that time we are going to discuss about the types of ribosome, what is the difference, etc., etc. As of now, 6 kingdom classification is based on ribosomal studies, and to be precise, which ribosome we are talking about, it is 16S ribosome. I think you all know these words, no? 70S ribosomes and 80S ribosome. The basic point of difference between a prokaryote and a eukaryotic cell. Prokaryote cell also has ribosomes and eukaryotic cells also has ribosomes, but the type of ribosomes is different, right? In case of prokaryote, what is the ribosome that we get it? Which type? 70S, and in case of eukaryote, is it only 80S? Both. So, if you say that it's only 80S, the answer would be wrong, because there are certain organelles in a eukaryotic cell where you see both 70S and 80S, and which organelle are we talking about? Kuntu organelle. Heard something called as semi-autonomous cell structure. Heard something called as another statement, endosymbiont of bacteria. Ei duta statement shunisan nai when we are talking about cell. Cell is a chapter where we are going to read about cell and the various contents of a cell, right? Organelles bilaka ami porhim. And there, while you are discussing 2 cell organelle, these are the 2 statements that you should learn. Endosymbiont of bacteria and semi-autonomous cell structure. Which organelle? Mitochondria and chloroplast, because these are the 2 organelles inside which you get 70S ribosomes and rest of the areas you see 80S ribosomes, right? So, that is the reason why we say in a eukaryotic cell, kenukwa dhoroner ribosomes thake, it is both 70S and 80S that is present in a eukaryotic cell. If you say it's only 80S, the answer would be wrong, right? But in case of a prokaryote, obviously it's only 70S, right? Okay, coming back to Karl Woese, 6 kingdom classification, studied what? 16S ribosomal structure. Third, he has given the concept of domain, right? What is a domain? I think you remember something called as taxonomic hierarchy. Living world, we have discussed about taxonomic hierarchy. What we know, that if you want to classify a specific organism, what do you need to do? You have to arrange it in a proper sequence. I think you remember that line, na? Keep pots clean, otherwise family gets sick. So if you are talking about that, highest ki asile? The highest category or the rank or the taxon is kingdom, and the lowest is obviously species, right? So according to the portion that we have studied, or according to 5 kingdom classification, up till there the highest taxonomic rank that we talk about is always kingdom. But it is only after 6 kingdom classification, or in the 6 kingdom classification, that something more than a kingdom was proposed, right? Normally kiman thake 7 obligate characters, right? Kingdom or pra species or okay, how many characters? 7, right? But something more than a kingdom. More than a kingdom means super kingdom, right? Instead of using a word called as super kingdom, what did Carl Wood use? He has used the word called as domain, right? So kingdom or upor ki thakibo? Domain thakibo. Next point, into how many domains were the organisms divided? Classification system always talks about the division or the placement of organisms, right? If you are talking about plants, if you are talking about microorganisms, or you are talking about animals, respectively they are placed into respective kingdoms. So according to this 6 kingdom classification, we come across a word called as domain. Now the next question is, what does a domain consist of? Since it has 3 domains, all the organisms that are present in this world are placed under these 3 domains, right? Hey, domain gita ki hoy, you need to know. So what is the question that is asked? First, who has given 6 kingdom? Second, it is based on ribosomal or 16S ribosome. Who has given the concept of domain? It's Carl Wood who has given the concept of domain. And what is the 6th kingdom? Whenever we talk about 5 kingdoms, what are the 5 kingdoms that is included? Monera, Protista, Fungi, Plantae and Animalia. So these are the 5 kingdoms. So tar upor jodi 6 kingdom classification ase, question kiya hai, what is the 6th kingdom? What did he do? He divided the Monera into Archaebacteria and bacteria. Before or prior to 6th kingdom, all these microorganisms were included under Monera itself. So what is the 6th kingdom? It is nothing but Archae or Archaebacteria. These are the points that you need to know. Likha? (16S) ribosomes, no? 16S nuhunilio. We come across 70S and 80S, no? What is S? Sedberg's unit. And what does it mean? It is also called as Sedberg's unit. Very good. Tako aru eta naam ase, no? It is also called as sedimentation coefficient, right? Why is it called as such? Why S? Sedberg was a person who has worked on that, and so it is called as Sedberg's unit. But you have a meaning for that unit also, right? That is nothing but it's talking about the sedimentation process. What is sedimentation? Sedimentation is another filtration, sedimentation, decantation, something that we have studied in our lower classes. Sedimentation means what? Settle down. What settle down? The heavier particles settle down. So something related with ribosomes. If you want to extract a ribosome from a cell, what will you do? If you want to extract mitochondria from a cell, what will you do? You will take the mitochondria, you will extract it. If you want to remove the ribosomes, is it possible to remove it individually? No. Why? Something related with the membrane. The cell organelle that does not have a membrane, ki asile? Ribosomes. So jetia at a cell organelle, it has a membrane, it's easy. Right? But ribosomes does not have a membrane, so it's not possible to remove it individually. So what they do is they go for a sedimentation process, right? They take the cell sap, they take the cell contents or the cytoplasm, and it puts the content in a centrifuge. Centrifuge means not ki hoy sedimentation hoy, and the higher or the heavier ribosomes settle down as compared to the lighter or less heavy ribosomes, and accordingly we have the sedimentation coefficient. I think you'll remember about the two parts of ribosome and all those. Eta ribosome hodai keita part thake. It's always two parts that it has. It has a dome shaped structure, it has a cap like structure, right? So based on that we have the word called as sedimentation. Okay. Right? What are the three domains? The three domains are archaea, bacteria and eukarya. If you are talking about archaea, what it will include? Only the archaebacteria. Bacteria boli kole the monera, and when we talk about monera, not necessarily there is just one type of monera, but we will be discussing about 8 types of monera, right? 8 type ase kintu 5 type he poribo lage. So all the monera or the prokaryotes, and if you are talking about eukarya, then obviously all the eukaryotes. And eukaryotes boli kole, it starts from which kingdom? Protista onwards. From Protista onwards, gutei bilak yat thakibo, right? And the last point that he will write, the 6th kingdom is clear? So up till here what we have completed is, we have completed talking about the contributions of various workers. What you need to remember, the different workers and along with their contributions, the books they have written, right? They were associated with which type of classification? The words we have already used it in the last class, natural, artificial and phylogenetic. So kunjon scientists kuntu classification are related, right? If you are talking about Aristotle, it's very obvious that he is related with which one? Artificial, right? Since, okay, we'll be talking about it. Is this over? Okay. Next part of the lesson would be kingdom system. So please put a heading, kingdom system. Okay, first is the two kingdom system proposed by Linnaeus. What are the two kingdoms? Plantae and Animalia. What did Linnaeus do? Right? While he was doing this classification, obviously if you have to classify, there should be a criteria for classification also. Kiba eta bostu sai pelai he organism took either plant buli koisile nohole animal buli koisile, right? So what would be the criteria? The best criteria that differentiates a plant and an animal is its movement, right? So the first thing that he has studied is, if we have observed the organism, the first thing that he has noticed is whether the organism moves or not. If it moves, he placed it into animal group. If it does not move, he placed it into the plant group. Second point of criteria was obviously nutrition. If it is photosynthetic, then it was a plant. If it is non-photosynthetic, then it was an animal. So there were certain criteria gita likha. Right? So, এইটোৰ ওপৰতো question আহে, you have to talk about this also. Okay? Sorry. Two kingdom classification obviously is not used any longer because it has like what improved, and now we have the 5 kingdom. So there should be a drawback because of which it was not accepted. So what is the drawback? Why was 2 kingdom classification not accepted? Eta homoyloke he accept korisile, ajikali to nokore, but there should be a certain reason, no? What is the drawback? Whenever we talk about the drawback, we have to talk about this line. It does not show any differentiation between eukaryotes, prokaryotes, unicellular, multicellular, photosynthetic, non-photosynthetic, right? কিন্তু কিছুমান যে তাত এক্সেপশনস আছে সেইখিনি কনসিডাৰ কৰা নাছিলে, ৰাইট? সো কিছুমান বস্তুটো এক্সেপশনস আছে, কিছুমান প্লেন্টসও আছে কিন্তু ফটোসিনথেটিক নহয়, ৰাইট? সো সেইখিনি বস্তু কৰা নাছিলে সেইকাৰণে। আৰু বোৰ্ড অফ নিউট্ৰিশ্যন বুলি কোৱাৰ লগে লগে ফ্ৰম দিছ পইণ্ট অফ ভিউ উই আৰ টকিং about the reason why the kingdom fungi got evolved বা why kingdom fungi was formed, because fungi কিহৰ কাৰণে ফৰ্ম হৈছে, কাৰণ প্লেন্ট লাইক হয়, কিন্তু in spite of that it is not photosynthetic. So according to the 2 kingdom classification, they did not have any idea the fungi nisna organisms bilak where will you put, right? We'll discuss about it. I think this part is over. Okay, next is another 2 examples that you have to talk about when you are discussing the demerits, right? Demerits porute, no, you have to talk about right? 2 kingdom classification did not have any idea where to put organisms like euglena, organisms like slime molds, etc., etc. What is the reason behind? Because whenever we talk about euglena, the first line that we remember, ki monot pore, what is euglena? Out of all the scientific information that you know after reading the chapter, that is different, but whenever we hear something, no, there is always a first point that comes to our brain, right? Plants puli kwa loge lege jeneke we remember photosynthetic, thik na? If you talk about bacteria, we remember that it is a prokaryote. So if you are talking about euglena, what is the first line that you should remember? Euglena forms a link between plants and animals because it has certain plant-like characters and it has certain animal-like characters also. According to the 2 kingdom classification, what did Linnaeus do? He has directly divided the organism, either it will be a plant or it will be an animal. But what about organisms like this, which show the characteristics of both the organisms, right? Similar is the explanation in case of slime molds also. Slime molds also show certain animal-like character and certain plants-like character. Slime mold monot ase nai porha? Slime mold under which category or which kingdom? We'll come to that. Slime molds or plasmodium, pseudoplasmodium, ebu words mon ase nai? Okay, we'll come. Okay, as of now, first ki likhiba euglena, first point likha shows a link between plants and animals. Nothing in details, this two lines from this chapter is more than enough. Details about euglena we will be talking about when we are studying about the kingdom called as Protista, right? Because euglenoids asile no, we will be talking about euglena at that time. Actually, there is no point of writing all these characters here, because once we go along the chapter, we are going to study in details about euglena and also in details about slime molds. Once you complete the details, it's very obvious that you will be able to take out the characteristics, right? So, if you are talking about slime molds, again it has an animal-like character and it has a plant-like character. Does anyone remember what is the character I am talking about? Because this is a chapter that you have already studied. Slime molds puli kole. Kenekwa type? Animal like you. What is the best character that describes the animal? It can move from one place to another, right? So, obviously, slime molds will move from one place to another. Because of that movement, it is put under the category of animals. What about plants? Why is it called as plants? Maximum character it has of that of an animal. But at one phase of the lifetime, it acts as a, or it shows a plant-like character. And to talk about a plant-like character, we are talking about the spore formation method, right? Spore formation, generally spore formation বা spore formationere reproduction korea boli koa loge loge, it is generally the plants that we remember, and especially fungi, right? Because fungi, we know there are so many modes of reproduction, but reproduction by means of spores is the most common one, right? So, similarly in case of slime molds also, during their reproductive stage, vegetative phase, they will remain like an animal, they will move from one place to another, they will go in search of food, etc., etc. But once they become reproductive, or during that reproductive phase, they show the formation of spore, and formation of spore is considered as a plant-like character, right? Animals are generally spore form no kore. Whenever we are talking about a plant, algae, we see spores, fungi, we see spores, even in case of bryophytes, pteridophytes, etc., we see the development of spores. So, formation of spores is obviously considered as a plant-like character. Clear? So, should I write it down or it's okay? Pariba ne likhibo lagibo? Pariba no? So, slime molds also both plant-like, comma, animal-like. I think that's more than enough. Because of these demerits, the two kingdom classification was no longer used. Next, we have to talk about the three kingdom classification. Yate no classification system bilak kune propose korise no, the one who has proposed it, always the question is asked about that, who has proposed the second two kingdom, who has proposed the third kingdom, etc., etc. Proposed by Haeckel. What is the third kingdom? It is Protista. Sorry. Whenever we talk about Protista, the line that we remember, it is the kingdom of unicellular eukaryotes. Right. So, two kingdoms, Plantae and Animalia. Next, three kingdom. What is the new kingdom we are talking about? We are talking about Protista, and this kingdom includes what? Unicellular eukaryotes, because we have already discussed within the two kingdom classification there was no differentiation between prokaryotes and eukaryotes, unicellular and multicellular, right. So, unicellular eukaryotes were totally taken in from that place and was assigned a particular kingdom, and that kingdom is called as Protista. Who has done it? Haeckel. So, A2 word important, A2 word important, A2 word important. This is all about the three kingdom classification. Next we have to talk about four kingdom classification. Who has proposed it? Copeland. And what is the fourth kingdom? Monera. Talking about Monera, what we remember? Kingdom of no unicellular. Prokaryotes, right? Yeah. Okay. Now we see two similar words. One is mycota and one is mycota. Right? So you have to read the question properly. Kihor upor thudise, mycota upor thudise na mycota upor thudise. Right? If you see a H, we are talking about Monera. Right? Jedi H nai, it is mycota, then it's the other name for fungi. Sorry. Five kingdom classification proposed by Whitaker, right? What is the fifth kingdom? Fungi, right? So even after the four kingdom classification, the placement of fungi was not clear, right? Fungi plants hoy ne, animals hoy, unicellular hoy ne, multicellular hoy, because if you remember there are examples of unicellular fungi and there are examples of multicellular fungi also, right? Normally in general fungi ki thake, unicellular organisms thake ne, multicellular organisms thake. Maximum of the fungi are, they are multicellular. One example of a unicellular fungi, yeast aru eta ase syncardium nalage hetu, kintu yeast is something that you remember, right? Maximum of that, if you are talking about all the basidiomycetes, all the ascomycetes, these are examples of what? Multicellular fungi. So 5 kingdom classification or ketaman criteria asil, there were 5 criteria for classification, and this is the question that is most frequently asked, right. So out of the 5 kingdoms you have to be precise. Question to keneke di diye, if suppose out of the 5 only one is there in the option, then it's very obvious that we will correct or we will mark that option, right. But sometimes what happen, all the characters or all the criteria are given in the option, and you have to choose which is the best criteria for this classification. What you need to remember is this classification was proposed to assign or to give a particular position to which group of plants or which group of organisms, fungi, right. Why was this classification conducted or done? Because fungi did not have a place to stay, right. What was so unique about fungi? It was not photosynthetic. It looks like a plant. It is plant-like characters, jeneke plant or keneka thake. It has a root system and shoot system. Almost thread-like structures is something that is seen in fungi. In spite of sharing many characteristics with plants, in spite of that, it could not prepare its own food. So basis or the most important criteria for the 5th kingdom classification ki hobo? Mode of nutrition, right? Our question maximum kya ho dise? Mode of nutrition of what, right? Difference ki? Phylogenetic relationship, it's always the evolution point that you are considering, right? I think you have heard something called as a family tree, right? So if you are talking about that, if you are drawing a family tree, if you are identifying the organism, that is a phylogenetic study. If you are talking about the ecology, means what? The role. Role means each and every organism in its ecosystem or in its habitat has a particular role. And which role? The 3 words that are most commonly used: producers, consumers, decomposers. So that is what is considered, right? So this was all about the kingdom system, right? Now the next part of the lesson, we have to talk in details about each and every kingdom, right? So after this, what we have seen is, according to the 5 kingdom classification, we have the 5 kingdoms, and 5 kingdoms, or suppose say ancient kingdom. Ki hoy? Ancient kingdom. Right? So what are the 5 kingdoms? Monera, Protista, fungi, plantae, and animalia. So since we are organizing or we are arranging these kingdoms in a particular sequence, ami to agote prokaryotes koa nai no, aba agote ami protista koa nai. What we have used this, or what we have said is, we have used the word monera first, and second is the protista. So obviously the sequence talks about something. It talks about what? It talks about phylogeny, right? So if you are talking about phylogeny, so we have an idea. There is suppose say primitive key, Monera, and most evolved, obviously Animalia, right? Now when we are talking about phylogenetic role, duta diagram akim, just see which one is correct. all the 5 kingdoms, right? Both the diagrams are talking about the 5 kingdoms itself, but as far as evolution is concerned, which is the correct diagram? A2? No, A2. This one. Why not this one? A2 kya nahoy? Plants cannot convert itself into an animal. Paribo niki? Adult plant hoy ase bole he turned into an animal. It cannot be that, right? So what does this sequence tells us? It tells us okay, Monera evolved hoise Protista. That's a correct explanation. From Protista we see the next evolved organism, that is fungi. Up till here it's correct. But you cannot say that all the fungi became plants and all the plants became animals, right? So this is not the correct diagram. Obviously this is the correct representation. Was there anyone who was asking me a question? Online students, kunbai kiba khudisla niki? Close ho gol. Huh? Ahille eta? Eta shuna pabo na pai tahate. Students, can you hear me? Online students? Okay. Doesn't matter. Chalo. Okay, so I was talking about this sequence. So from this sequence, what we understand, primitive organisms, Monera a little bit evolved. We had the Protista. Protista is the only group which has diverse forms of organism. This is one important line that you need to remember. Out of the 5 kingdoms that we are talking about it here, each and every kingdom have their respective members. When we are talking about the kingdom Plantae, what does it include? It includes only the plants. If you are talking about Animalia, what does it include? It includes only the animals. Similarly, if you are talking about Monera, only the prokaryotes. If you are talking about Protista, only the unicellular eukaryotes, right? But if you are talking about Protista, another point that you need to remember, this is the one kingdom that has maximum numbers of diverse organisms. It is not only plant-like protists that is present, it is also animal-like protists that we see in this kingdom. We also see fungi-like protists also. So if you remember, you have read about Protista, tinta category porisila, right? Fungi-like protists, plant-like protists and animal-like protists. Remember the words called as dinoflagellates, diatoms, euglenoids, right? And if you are talking about Animalia, animal-like protist, right? Certain examples of protozoans, those are animal-like protists. We'll be talking about just few lines about it. This too, we are going to do it in details. In this section or in this kingdom, maximum question has been asked about the plant-like protists, which is divided into 3 categories: diatoms, dinoflagellates and euglena, right? What is fungi-like protists? The example that we have discussed a little before, slime molds, right? So slime molds ki hoy? Protist, kar nisna dekhat, fungi nisna dekhat, and fungi nisna dekhat boli koa loge loge. What is the first character that we remember? Saprophytes, right? Because fungi are always saprophytes, right? So this was all the information. Now we'll start with the kingdom Monera. Heading koriba kingdom Monera. Okay, let me write down the points first, right? Tar pisot ami explain korim. So please write the points about Monera. Each and every point that you will write is important, right? We have not written any point that is not important or it is not asked. Egithi bhalke poriba. So the organisms that belong to this kingdom are considered as the most ancient organisms, and when we use the word most ancient, kiho karone use koru? Because these are the organisms that were present many years before also, that year 3.5 million years ago, right? According to Whitaker, it is the kingdom of prokaryotes. As far as nutrition is concerned, so the organisms could be autotrophs, they could be heterotrophs also. And autotrophs, heterotrophs, I think we understand who do the organisms that prepare their own food or do not prepare their own food. If the organism does not prepare its own food, then from where will they take the food? Other organisms, and depending on that other organism, we have different categories of heterophytes, because the host from where they will take the food might be living or it might be dead also, right? So accordingly, we have categories of heterophytes. So next is, give an example of an autotrophic monera, give an example of a heterotrophic monera. Very good. Cyanobacteria, also called as blue-green algae. And heterotrophs. Now, when I am talking about bacteria, obviously it has certain exceptions. I think you have heard photosynthetic bacteria. Kisuman bacteria photosynthetic hoy no hoy, right? So, but as a whole, the group bacteria is a heterotroph. Apart or except that photosynthetic group, maximum ki hoy heterotrophs hoy. We understand the meaning of ubiquitous, understand the meaning called as universal, right? It's the same meaning. Universal mane ki? Present everywhere. Ubiquitous, found everywhere. And by the word everywhere, exactly what we are understanding is these sort of organisms could be found almost in all the places. It might be a very cold area, it might be a very hot area. I think you have heard something called as a hot spring. Right, hot spring or thakibo pare ba bahut thanda jaga to thakibo pare, right? Next is they could be found also in the body of the organism or on the body of an organism. Organism to upor to thakibo pare ba it could be present inside the organism also. And when we are talking about a type of a prokaryote, the best example that we talk about one that is present inside the body of an organism, we talk about E. coli. E. coli ki hoy bacteria. So if it is a bacteria, so obviously it's a prokaryote. So we have certain examples of prokaryotes which are found even inside the body of a human being, right? And since they are found everywhere, we are using the word called as ubiquitous or widespread, right? Yet the explanation halaki one word is sufficient. এটা বস্তু লিখি দিয়া less known organisms. Okay, they are ubiquitous, but in spite of that, we generally say that they are less known organisms or less known kingdom. Why we are using the word less known? Because there are so many other prokaryotes which has not yet been discovered. Right? Etiao bahut ase junkhini ki ami najanu, right? Talking about cell wall present or it might be absent also. Give an example of a monera where will you see the cell wall. Cell wall thaka eta monera example dia, without cell wall eta monera example dia. The hint is the smallest microorganism ki hoy. The smallest microorganism with a cell wall, the smallest microorganism without the cell wall. Smallest organism boli kole, it is always what? Microorganisms with cell. So, smallest microorganism with the cell wall, bacteria. Without the cell wall, mycoplasma, right? Mycoplasma is also called as dash of the plant kingdom, joker of the plant kingdom, joker of the plant kingdom. Why they are called as jokers? We will be talking about it. I mean, all of this are for him. Okay, before writing another question, whenever we talk about cell wall is a characteristic that differentiates a plant cell and an animal cell. That is the basic characteristic. If a cell wall is present, it's a plant cell. If a cell wall is absent, it's an animal cell. Next, talking about cell wall, what is the composition of cell wall? If the question is asked, plant cell or cell wall or ki thake? What is the basic component? Cellulose. If you are talking about the fungal cell wall, then what is the word that you remember? Chitin. If you talk about the bacterial cell wall, what do you remember? Something that starts with what? P. Yeah, peptidoglycan, that's the best thing, or right? Peptidoglycan boli nokoi pelai, peptidoglycan that is correct, peptide form or glycan form, hekhini ami kom, kintu generally ami polysaccharide boliu koi thoi dibo paru, right? Kintu peptidoglycan is the best word. আমি অলপ পিছত পঢ়িম হেতু ওপৰত. So this is all about the cell wall. Next point is about locomotion, right? The members of this kingdom. What do you know about locomotion? One monera that does not show locomotion. Cyanobacteria, right? Can't have moved no correct. What about reproduction in case of prokaryotes? Kenuka dhoroner reproduction dekha pao? Reproduction broadly divided into what? Sexual and asexual. So prokaryotes ot ami ki dekha pao? Asexual. So they will mainly reproduce by asexual. Is sexual reproduction present or seen in bacteria? Sexual reproduction dekha pai na pai? Sexual reproduction is a characteristic of higher plants. When will you say an organism is undergoing sexual reproduction, or what is the basic necessity to undergo sexual reproduction? Formation of what? Gametes. So if gametes are formed, then obviously we say that sexual reproduction hoise. Jedi gametes form kora nai, tetiale sexual reproduction hoa nai. That is one explanation. Second, what happens in sexual reproduction? We know gametes are formed. What happens to the gametes? The contents fuses, right? So if we say the contents fuses, so what we are saying is the genetic material gets fused, or there is an exchange of genetic material. So in case of bacteria, We do not see the formation of gametes, but we see the exchange of genetic material. Are you understanding the statement? We do not see the formation of gametes, but we see the exchange of genetic material. So, we cannot be using the word sexual reproduction. Instead, we use a word that is called as sexual recombination. Right? So, bacteria, asexual reproduction is present, sexual reproduction is absent, but sexual recombination is present. And while talking about recombination, I think you have come across the words called as conjugation, transduction and translation. Right? Conjugation that we'll discuss. Right. So, ultimately mode of reproduction Our asexual mode boli koa loge lage. What is the most common method of asexual reproduction? Binary fission. Another one, spore formation. Bacteria spore form kore ne nokore? Bacteria generally the most common method is binary fission. So, spore formation, as I have said, no, spore formation is generally related with the next group of plants, fungi, plant, tea, etc., etc. But in case of bacteria also a specific type of spore we need to talk about. A type of a spore that is formed in a bacteria during unfavorable condition. Favorable condition they will never form a spore, but during unfavorable condition they will form a spore, a particular spore that is called as the endospore. We will be talking about it. As of now, if you are talking about reproduction in case of prokaryotes, it is mostly asexual, binary fission and spore formation, and if you are talking about sexual reproduction, it is absent. Yeah, ebilakor pora question ahe kintu dey. One monera that lacks locomotion, one monera that shows locomotion, one type of a monera that is autotrophic, one type of a monera that is heterotrophic. So, please read the points properly. Right? Right. So, types of monera, it is not only bacteria that belongs to monera, but we have other organisms also, right. So, monera has 8 types. These are the 8 types. Out of these are the 5 types that you need to study. 18 tar upor questions aha nai. Right. So, details পঢ়িব লাগিব পাঁচটাৰ ভিতৰত. Out of this maximum details, eubacteria. What is a eubacteria? A true bacteria is called as a eubacteria. Why are we using the word true bacteria? Because there are certain bacteria that are cyanobacteria also. Those are not true bacteria. right? Eta bacteria boli kole, they will have certain type of characteristics which will differ from the group called as cyanobacteria. Cyanobacteria is also a bacteria, right? But if you say a true bacteria, it should have the characteristics that differ from the cyanobacteria or maybe from archaebacteria, right? Talking about these points, one line that you have to remember, archaebacteria, the most ancient type of bacteria, a group of bacteria that can survive in unfavorable or extreme environmental conditions, right? Any type of a extreme environmental condition, whether it might be very hot, whether it might be very cold, or maybe I think you have heard something called as rumen of cattle, methanococcus, gobar gas, etc., marshy area, right? So this is the group that is associated with that. Next question is, why is it like this, that this group can stay in extreme environmental condition? Because of their unique characteristic of a cell wall. Next point of difference is, what is so specific, or what is the difference between the cell wall of archaebacteria and a true bacteria? Because talking about eubacteria, obviously details will do. Talking about cyanobacteria, blue-green algae, talking about blue-green algae, the first word that comes to our mind is soil fertility. When we talk about soil fertility, the next word, nitrogen fixation. The third point, the common examples of cyanobacteria, nostoc, olocera, etc. Tinta hole, mycoplasma. Mycoplasma, as I said, a microorganism without a cell wall, the smallest creature on earth. Next, many diseases that are caused by mycoplasma. You have to study the name of the diseases. Mycoplasma can cause disease in plants, animals, and also in case of human beings. So question ahe, identify the disease that is caused by mycoplasma in case of a plant. Or maybe after the end of this chapter, as I said, no, you have to study about bacteria, you have to study about fungi, you have to study about mycoplasma, and also about virus. All the four categories have their respective infections. Viral disease was a, bacterial disease was a, fungal disease was a, all type of disease. So at the end of the chapter, the question that is asked is, eta disease didibo, right? And you have to identify which type of a disease is it, whether is it a viral, or is it a bacterial, or it is a fungal. So obviously disease part you need to remember, right? Next, mycoplasma, okay. Coming to again another line, the joker of the plant kingdom. Why is it called a joker? Because it can keep changing their shape and size because they do not have a cell wall, right? Next is actinomycetes. Actinomycetes, they are also called as ray fungi. So this is a group that looks like fungi. We are using a word called as ray. What is the meaning of ray? They have elongated cells, or cells that we use the word called as filament. Normally when we talk about bacteria, what is the shape? Bacteria shape ki? Bacteria tabuli kole, what is the idea that you have? Heard those words, no? Coccus, bacillus, some rod shaped and all those comma shaped, etc., right? But you have a certain type of bacteria that is like a filament, and when we are using a word filament, obviously filament eta kenike form korbo, cell bilak etar logot eta attached hobo laibo, right? One cell should be attached with the other, and ultimately they will form a filament. If they have a structure like that, that is what is called as filamentous, right? So the question that is asked is, identify the filamentous bacteria. So filamentous word to hunile, you have to remember actinomycetes. Second point, bacteria causes diseases, right? Actinomycetes also cause diseases, we will be talking about. Next, when we talk about microorganisms, we also remember the production of antibiotics. Antibiotics kune produce kore? Maximum bacteria, fungi, ebilake antibiotics produce kore, microorganisms they produce. But you will be surprised to know that maximum of the antibiotics kune produce kore se actinomycetes. So you have to remember the antibiotics that has actinomycetes origin are antibiotics that has fungal origin, antibiotics that has bacterial origin. So this is under the three categories that you need to study, right? So we'll be talking about the first one, that is eubacteria. So heading koriba eubacteria, archaebacteria later. Right? So if you are going to study this, what are the contents of this topic? You have to talk about Bacteria itself, kenikwa dekhat, cell to kenikwa, cell shape, cell structure, etc., etc. Next, you have to talk about its method of locomotion. You have to talk about the organs for locomotion, that is flagella in precise, like the structure of flagella. You also have to talk about the reproduction and also nutrition in case of bacteria. So obviously, eubacteria is a very longer topic. We will be starting with the type of the bacterial cell or the diagram. Right. Whenever we talk about a cell, either it is a prokaryote or it's a eukaryote, no? Cellot ki porisa, the structure of a prokaryote is slightly different from the eukaryotic cell. Both of them will have two different diagrams for that. Here what we are going to do, we are going to draw the bacterial cell, and ultimately this portion is going to include all the information about a prokaryotic cell. So in the chapter cell, we are not going to discuss again the prokaryotic cell. That we can discuss the structure of the eukaryotic cell, because prokaryotic cell is something that we are going to discuss it now, right? Okay. Talking about the bacteria, the most common type of bacteria ki hoy? Bahut bilak shapes ase, no? What is the most common type of bacteria? Which shape? Rod shaped, right? So we are going to draw a bacteria. Please draw it down. It's a big one. ডাঙৰ কে নাকিলে পইণ্টছ লিখিব দিগদাৰি হ'ব instead of writing the points so you can mark it in the diagram and write it down, right? Just a rough diagram, you don't have to be Leonardo da Vinci, right? Just a rough diagram so that you can understand. These are nothing but hair-like structures. a rough diagram. So, the structure of a prokaryotic cell, right? Cellot aru ami prokaryotic cell nupodhu. Etu pohile, it's all the parts of a prokaryotic cell is something that we are going to discuss here itself. So, because if we talk about prokaryote, it is always the bacteria as the best example that we remember. Okay, so is the diagram over? Another point that you can write is capsule, cell wall, cell membrane ase no, ei tinta eneke include kori cell envelope. Ekdom last to capsule. Mane soap bacteria re cell wall aru cell membrane থাকিবই কিন্তু capsules এ থাকিবই not necessary কিছুমানৰ capsule নাথাকিবও পাৰে capsule থাকিলে advantage কিবা এটা আছে নাথাকিলে disadvantage এটা আছে we'll be talking about it. Okay, about the points about this diagram, right? মই যেতিয়া কৈ যাম তোমালোকে লিখি যাবা. Right, instead of me writing it everything, okay, as I keep on saying, just important points that you have to talk about. You don't have to write everything in details. Okay, I think this is over now. We can explain. Look, if you are talking about a bacterial cell, the rod-shaped bacteria is the most common type of bacteria that we see in nature, and so this is a diagram for rod-shaped bacteria or the bacilli group, right? Okay, in a bacteria, what are the parts, or in this cell, what are the parts that you see? First, talking about this something called as the cell envelope. Cell envelope consists of three parts, that is capsule, the cell wall and the cell membrane. Obviously, details about the cell envelope in the next class. ঠিক আছে cell wall খন কেনেকুৱা দেখাত, how is it made of, what are the things that is present, we have to talk in details. As of now, what you need to remember, in a bacterial cell, we see something called as the cell envelope. The cell envelope consists of three layers, capsule, the cell wall and the cell membrane. First point about the capsule. The capsule might be present or it might be absent also. Not necessary each and every bacteria will have the capsule. If a bacteria has a capsule, it provides a virulent character to the bacteria. You have to understand the meaning of virulent. Eta bacteria jodi capsule thake, tetiahole ki hobo? He bacteria to extra protection eta pabo, right? Extra protection means what? Its genetic material cannot be destroyed easily because it has a third layer, right? So, if a bacteria does not have a capsule, it is more prone to get killed and it would be non-virulent or non-infectious. If it has a capsule, then it will be virulent or it will be infectious. Why? Because it has the property to survive, and plus if it survives, it will cause disease. So, the most important point about a capsule, if present, virulent, if absent, non-virulent. Next, if a bacteria does not have a capsule, instead of a capsule, what it has something called as a slime layer. Next class, we are going to write about slime layer, right? So, capsule kon jodi nathake, you see a slimy layer of substance that is present. It makes the bacteria very sticky and like slippery. Slime layer, no? So, it's obviously slippery. What is the disadvantage of a slime layer? Right, if it has a capsule, it gives the extra protection, genetic material gets preserved and it is virulent. If it has a slime layer, this slime layer could be removed very easily even with just washing it with water also. You take the bacteria, wash it with water, the slime layer vanishes, and if the layer vanishes, it is easy to kill that bacteria, right? Next, so this was few points about the cell envelope, details in the next class. Next is certain granular structures could be seen inside a cell. Two types of granules, one is called as the volatile granules and the other is called as the glycogen granule. What you need to study, what is the content? phosphate ক'ত থাকে, carbohydrates ক'ত থাকে. Food containing granules is called as glycogen granule. Maximum question has been asked about this one. Next, talking about the nucleoid, right? Nucleoid or the genetic material. Obviously, prokaryotic cell does not have a nucleus. It does not have a nucleus because it does not have a nuclear membrane. If it does not have a nuclear membrane, genetic material থাকিব না থাকে. থাকিbo, right? So this genetic material that is present in the form of a circle, so it is called as what? Circular DNA, the type of genetic material in case of a prokaryote, circular DNA. Talking about histones, have you heard the word histones before? DNA packaging, right? So histone protein, something that is present in case of a eukaryote, absent in case of a prokaryote, right? So this is all about nucleoid. This is a genetic material. Apart from this genetic material, it also has an extra genetic material, that is what is called as a plasmid, right? What is the most important function of plasmid? Plasmid আমি কেতিয়া পঢ়িম, if you remember something called as conjugation, as I said, there is a sort of genetic recombination or sexual recombination that is seen in case of a bacteria, right? We have a process called as conjugation. Who helps in conjugation or what gets transferred? It is the genetic material that gets transferred between two bacteria. Which genetic material, to be precise? A part of a plasmid, right? We'll be talking about it in details. Next is cytoplasm, we understand. Okay, cytoplasm details in the chapter called as cell. Talking about granules, we have talked. Another structure, vacuoles, right? Vacuole is a characteristic of what? A plant cell, because we see a big vacuole in the center itself, and that is the reason why the nucleus is pushed to the periphery, right? In case of an animal cell, generally we don't see vacuoles or very less in number. In case of a bacteria, vacuoles are generally absent, but we have a type of vacuole that is called as pseudo vacuole. What is pseudo? False vacuole, right? Another point, is vacuole present in bacteria? No, vacuoles are absent in bacteria, but pseudo vacuoles are present. In which type of a bacteria will you see the pseudo vacuoles? It is seen only in aquatic forms, because pseudo vacuoles of the vacuoles are also called as what? Gas vacuoles. Obviously, it will have air, and if it has air, buoyancy or floating or help koribo, right? Next, from the cell envelope, you see certain hair-like structures that is coming out. What are the, or how many types of hair-like structure? 3 types of hair-like structure. You have the flagella, you have the pili, you have the fimbriae, right? Out of these 3 hair-like structure, it is only one structure that helps in locomotion, and the rest 2 does not help in locomotion. Question ahe, does pili play a role in locomotion? No. Does fimbriae play a role in locomotion? No. It is only the flagella. Talking about flagella, again details about flagella in our next class or maybe next to next, right? Flagella, from where does it arise? What is the structure of the flagella? I think you remember hook, the basal part and the filament. Tinta part asil no? Okay, corpora arise korise. You have to see the diagram properly. It is something that does not arise from the capsule. Capsule opera arise kora nai kintu. If you are talking about this two, corpora arise korise, they have arise from the capsule. That point of difference you need to identify. Next is we have finish. Okay, a very important structure called as mesosome, right? What is mesosome? It is nothing but an invagination of the plasma membrane or the cell membrane, right? What is the meaning of invagination? Humai jai, right? Ekon membrane eneke ase straight. He said bhitorle humai jai, right? That is koi ki koi. Invagination. Invagination hole, what is the advantage? If a membrane is like this and if a membrane protrudes inside, there is a difference, no? What is the advantage? Something about the surface area, right? So in this case, the surface area increases. Not necessarily a bacteria will have just only one structure like this. It might have many structures also. This structure could also be compared to a structure that you see in an organelle in case of a eukaryotic cell. Mitochondria. Mitochondria eta bostu ase that is called as cristae. Remember the cristae, that folded invagination from the inner membrane of mitochondria. What is the work of a cristae? Mitochondria kam ki? It is called as the powerhouse of a cell. Why is it called as the powerhouse of a cell? Because ATP is produced. Where exactly is ATP produced? Mitochondria is a big structure. It has 2 membranes, it has a matrix, it has a lumen, it has a cristae. ATP production hoy, ATP production coat hoy, and what is the requirement for ATP production? Inner membrane or inner membrane ATP compound to inner membrane dekha paba ne bhitor dekha paba? Obviously inside, right? It is something that is present in the cristae that helps in the ATP formation. Remember something called as FOF1 particle, right? Kot thake crystal. FOF1 particle helps in the formation of what? ATP, right? Because ATP formation always requires two things. What are the things? Concentration different, proton concentration, aru at an enzyme, ATPase. Where is that ATPase found? Precise. FOF1 part, right? Exactly where? F1? No, F0. F0 is something that has 0 inside. F1 part, right? F1 is the head part, F0 is the tail part. So what do you remember? If it is 0, it does not have anything, right? So F1 not ki thake, it has an enzyme. That enzyme helps in the formation of ATP. Now that is something related with respiration. Eukaryotic cell, when we are talking about respiration, we remember the mitochondria. Prokaryotic cell also respires, but they do not have mitochondria. So where will they respire, or where will ATP form, or where is respiration conducted in a prokaryotic cell? It is always in the mesosome. So what is the function of a mesosome? To conduct cellular respiration more efficiently in a prokaryotic cell. Clear? Function of a mesosome? Respiration. Function of a mesosome? ATP production in case of a prokaryotic cell. Talking about mesosomes, mesosomes are of two types. We see two types of mesosome. Duita mesosome, obviously they will help in ATP formation, but one type of a mesosome that is very nearer to the genetic material is called as what? Septal. This is called as what? Lateral. Lateral means what? Something that is present in the periphery or in the side, that is called as lateral, right? This mesosome which is present near the genetic material is called as septal mesosome because it helps in the septa formation during cell division. We have a cell, from that cell two daughter cells will be formed. If from one cell two daughter cells needs to get formed, there should be a cell wall or a boundary or a septa. So someone should form that septa. Eta cell duta bhagot bhag hoy goise. Bhag hole to he boundary to form koribo lagibo no. Hey septa to who forms the septa? It is the septal mesosome that forms the septa. So another function of a septal mesosome helps in the process of cell division, helps in septa formation during cell division, right? But out of these two mesosomes, maximum question has been asked about this and not about septal. So the next time you hear about mesosome, always remember the most important function is ATP production or cellular respiration. Clear? Okay. The last point about this hair-like structures, we understand flagella, locomotion, pili and fimbriae. Ki kam? Jedi conjugation monot ase. Attachment, right? If one bacteria, this is another bacteria, they have to exchange the genetic material, then they have to stick together also, right? If they stick together, then only the genetic material will get transferred. So this sticking purpose, kune koribo, it is the pili. So pili and fimbriae, main work, attachment, no role in locomotion, flagella, only the locomotory organ. This is all for today. In the next class, we'll continue. Thank you. PDF you have got, right? You have received a PDF of this chapter, right? So what you can do today is you can go home and just compare the portion that we have studied, right? This is the portion that is already there in the PDF. If certain points that you did not write it down in the class, PDF for yes, and we'll continue from here in the next class. Next class one, today is Sunday. Tomorrow we have a class that is organism and population. Tuesday we have morphology. Wednesday.